right. So how are you guys doing today?
Good.
Friday. So you will be doing good in about 4 hours. Okay. I hear you. So today we're going to be going over essential amino acids, non-essential amino acids, and then how we make all the non-essential amino acids. So who can tell me really quick what makes an amino acid essential?
We cannot synthesize it.
We can't make it. And so since we need it and we can't make it, we have to get it from food. So what makes an amino acid then non-essential?
We can synthesize it.
We can make it. And so today, a lot of what we're going to be talking about is how we make all those non-essential ones. All the essential ones, you just need to know them, and then you're good. The non-essential ones, it's like, how do we make those? What pathways can be broken? And therefore, if the pathway to make them is broken, then all of a sudden they become either essential or there's some other disease state associated with them. And that's why we want to know about the pathways. So let's go ahead and start with how we can tell essential and then non-essential, which I'm not going to actually write a list for really quick. There is a helpful acronym for the essential amino acids that I find myself rather attached to. You may have seen this, you may not have, but private Tim Hall. And so just like a private in the military, and his name is Tim Hall. So for the P here, we have Prolene, then we have Valine, then we have tryptophan, then we have threonine, then we have isoleucine, methionine, histidine, we have arginine, and then we have lysine and leucine. Now I'm going to go ahead and circle here a couple, because with private Tim Hall you can kind of be like, histidine, and it'll eventually come up. But there's two of them that there's more than one A amino acid. And so you'll want to be sure and know that arginine, you have the pirate. Apparently private Tim Hall has a spotty background. So arginine, and then there's one of the T amino acids that is missing, and that is tyrosine. Okay? And so just notice that, and I'll go ahead and write tyrosine down here at the bottom because it is the one that is not tyrosine. It is the one that is not included. Okay? Does that make sense?
Tyrosine is not included in this, but it is still essential.
It is non-essential.
It is non-essential, but it can become essential if we don't have.
Yes, we're actually going to go ahead and talk about conditionally essential, which is where tyrosine fits. So we have, and I'm actually going to shift where I wrote tyrosine over because it doesn't make sense to write it underneath the essential.
What did you say again about arginine? Sorry.
Arginine is the letter A in private Tim Hall. There are other A amino acids like alanine, but those are not the ones in the essential. So just arginine is the letter A, and so I wanted to highlight it because then it's like private Tim Hall, got it down. Now you're on the test. Now it's like, is it arginine, alanine, or great, fantastic, my favorite. All right. So conditionally essential. We have 3 in particular that fit here, and I like to remember them again with an acronym. With this, it's about finding a way to remember because there's just a lot of information. And I remember it with titch, like I'll be with you in a titch, okay? And so there's a lowercase y there. So this one is going to be the tyrosine that we saw earlier. Tyrosine. And then this next one is going to be a C, and all of these, the second letter is a Y. So it's Ty, Cy, Hy. Cy is going to be cysteine, spelled in whatever way you prefer. There we go. I like that today. Okay, I know it's wrong.
Is it?
It is. There should be an E in there somewhere, and I'm not going to commit. All right, so tyrosine, cysteine, and then one of our not one of our main 20 amino acids, but she wanted you to know this one in the slides that I had was hydroxylysine. And I just heard that there's in the slides also a hydroxy, and so we'll have hydroxy have two. Hydroxyproline. Okay, so tyrosine and cysteine, they come directly from amino acids. So, whoa, okay, I lied. Thank you. If anyone ever catches me doing something like that, please just yell it out. Apparently there's more than one P that I didn't think about. I was trying to highlight all of them. The first P in the essential, going back to the essential, is phenylalanine. And you'll find yourself double checking that as you write out the conditionally essential, because that's where tyrosine comes from, which we talked about last time. You remember phenylalanine, and then dehydratase or dehydratase, and then that made tyrosine. All right. Next, cysteine comes from methionine. And then for hydroxylysine and hydroxyproline, those make sense. They come from lysine and they come from proline.
But we don't have proline on our...
Prolene will be in the non-essential. So everything we haven't written is going to be non-essential.
Okay. The last two come from lysine.
This one is from, so it's hydroxylysine, and so from lysine, hydroxyproline from proline. And I haven't looked at pathways for those ones, so that's from memory. So there you go. All right, so something that I think would be helpful is to make sure that you have the essential ones down, because that's probably going to be a question in some form. Go ahead and see if you can get the essential and then the conditionally essential, and remembering especially the P and then the A, and then which T is not in the essential. Does that make sense? Okay, go for it.
What was the PV again for your...
Private PVT.
Oh, private. Okay,
Tim Hall.
Why did you write the non-essential one there?
Oh, because I write stuff in weird places sometimes.
Oh no, I was just curious why.
So,
Oh,
right there, conditionally essential will become essential if you don't have what makes them.
yes, right. Thank you. The cysteines derived from methionine?
Yes, cysteine from methionine, tyrosine from phenylalanine, and then hydroxylysine and hydroxyproline, I'm pretty sure come from lysine and proline. From memory, that's what serves, and I didn't see it in the slides when I went through. I think it's one of those things that she just says as she goes because it's in the name. But, all right. And so now we get to move on to how to make a lot of other stuff. So let's start with the two guys who can carry two nitrogens. What do we have that can carry two nitrogens? Glutamine, and then I thought I heard, I didn't quite hear.
Asparagine?
Yes, asparagine is the other one. So glutamine and asparagine. And I like to have these two side by side because their making and breaking has a lot in common with each other. And so, let's see. Which one was enzyme? Pink? Okay.
Thank you.
All right, so to make glutamine, you're going to make it out of glutamate. And you will use the enzyme glutamine synthetase. To break glutamine, you will use the enzyme glutam, basically glutamine ACE, glutaminase. So, to make asparagine, you're going to have one of the unique steps. Alright. Okay, it really is. Alright. This is going to be asparagine synthetase. And then for breaking it down, you're going to have, what do you think it would be named? Asparaginase, exactly. Asparaginase.
That's asparagine synthetase.
Yes. And what was the question? That was going to be the question? All right. Okay. So those really are just, they're both named after the two nitrogen compound, glutamine, asparagine, and you just say synthetase or break it down, just ACE, glutaminase, asparaginase. It's really nice. All right. So who remembers when glutamate is has a nitrogen removed, what do we call it? Alpha ketoglutarate. Excellent. And for aspartate, what will it become? All the animals like to be together. Good guess? Yes, oxaloacetate. And so you have donkey, which is ass. It's a word for donkey that is not rude when it's a donkey. All right, and then you have oxaloacetate. And so you have an ox. And those three go together. All right, and now here is where we have a slight difference. So glutamine to glutamate, alpha ketoglutarate. Between glutamate and alpha ketoglutarate, we're going to have a glutamate dehydrogenase. Catalyzing that reaction. Whereas for aspartate, if you guys remember, which was one of the king of transporting nitrogen in the blood? Glutamine. Yeah, occasionally you see other ones in the blood, but glutamine does a lot of it. And so we try and get, when we're dealing with nitrogen, we're trying to get them all to go into the glutamine, glutamate, alpha ketoglutarate kind of situation. And so when we're breaking down asparagine to aspartate, it makes sense that the predominant enzyme would be one that shifts the nitrogen over into the glutamine family, the glutamate family. And so instead of just a dehydrogenase that basically throws the nitrogen off into the wind, it's going to be a glutamate aspartate transaminase, or aspartate transaminase, AST. Aspartate transaminase, and I'll write it out.
That acts on the glutamate to make alpha ketoglutarate, is that what you're saying?
It takes the nitrogen away from this guy
And adds it.
to an alpha ketoglutarate to make a glutamate.
Perfect.
So this is all one enzyme. I just put the acronym at the top because you will see AST in blood work. And so, oh, you have high AST. That means you probably have some damage, liver or muscle, and so you want to know when that's high. All right, so this is just taking a nitrogen over, so I'll write that in not pink since that's not an enzyme.
So they said transamination.
Yes. Yep, it's just another transaminase, and it's named pretty much the same way as most transaminases. So, some kind of amino acid transaminase. It's our favorite naming family because they tend to be pretty consistent.
So the transaminase, I'm putting like that enzyme is going with an arrow up to glutamate because it adds the nitrogen to the ketoglutarate, making it glutamate, right?
Yeah.
So then you get an arrow going.
Yes, exactly.
We can go ahead and have it
that's
go like that.
Yeah, okay, cool.
And we'll add this guy to that, and then that makes it nice.
So adding aspartate,
The nitrogen.
add the nitrogen to aspartate to glutamate.
No, aspartate is donating a nitrogen to alpha ketoglutarate to make glutamate.
Oh, okay.
And
Right.
That's how you get the solo acetate.
So the nitrogen's going
Right.
this way.
Yep. So that's a two-part thing. So it makes glutamate and it makes oxaloacetate. So in order to
Right. It does both.
Cool.
So shouldn't the nitrogen be going to the alpha ketoglutarate?
It does. The alpha ketoglutarate and the nitrogen are meeting right there, and they make glutamate in the way that I drew it.
I did it like it goes to the alpha ketoglutarate and that goes to glutamate.
Yeah.
So these two.
Good deal. All right, so go ahead and see if you can quickly go ahead and name the top half of the pathway, what the molecules are, and then what the enzymes are.
Can you say the enzymes real quick for me one more time?
Yes, so I'll go ahead and run through it really quick. So you have glutamine and asparagine. That's going to be made with glutamine and asparagine synthetase. And then both of them will be broken down using glutaminase or asparaginase. Then you end up with glutamate and aspartate. Then you can name their pathways. That's going to be with glutamate dehydrogenase, or the funny one with aspartate transaminase, which will send the nitrogen over to the alpha ketoglutarate and make glutamate.
Perfect.
So the nitrogen is going to alpha ketoglutarate.
Yes. I can adjust the pathway since that seems to be making more sense to people when it's.
It's not synthase, it's the synthetase, right?
Right. Both of them are synthetase, which tells you that it takes ATP.
And the dehydrogenase, is that the one that requires a
Yes. Dehydrogenases have to have somewhere to stick the hydrogen.
Right, okay. So that,
Or take it.
So they reduce NADPH? So you have to have NADPH.
Reduce or
NAD,
oxidize?
Or NADP. NADP is right.
One of the two.
One of them. Oh, that's right. Okay. Accountable for it, just knowing that it needs.
It's the small things.
And
Thank you.
It needs the H though, right?
Exactly.
NADH, NADH or NADPH?
It needs.
NAD. Or NADP.
So yeah, so you're gonna take, I believe that it is NADPH or NADH, and you're adding the H, and therefore you're reducing to make it into ketoglutarate. You're putting one more hydrogen on, but I'm not gonna swear that that's the direction it goes.
Yes, you're right.
It makes sense because it would
It's a
replace the nitrogen and bump it off with a hydrogen. Because then the nitrogen can go away and leave the hydrogen in its place. Okay. So, there was that couple that I feel are nice together. So next, let's go ahead and do a nice easy one. Alanine. Yay, pyruvate. Pyruvate. All right, we can just have kind of like 2 exclamation points. They're super happy together. And that's about all that they do is, there you go. So, yeah, this is,
Well, what do we need to know?
for that, I mean, the enzyme, there's like
Was there anything?
2 different enzymes that can be used for it, and which one does she want you to have here? She doesn't even name it. Okay, she doesn't care. It goes between the 2.
Okay. Just the alanine. They can go back and forth. Okay.
Yep. This is part of the Cori cycle. If you remember, the muscle would make alanine, it sends it off to the liver, and then the liver will take alanine and turn it back into pyruvate, and that's how you get energy out of most amino acids, new glucose, I should say, gluconeogenesis. That's the favorite pathway of gluconeogenesis from muscle amino acids.
Okay.
Cori cycle. So I'll put that as a little note, Cori cycle, if you want to go and...
That's okay.
When I added the word want in there, if you want to, I should
Right.
do that.
She had broken them together, alanine and aspartate together.
Yeah, they both use similar enzymes. I preferred putting these 2 together because they actually have some interplay, but
Okay.
similar enzymes will work with alanine and, like right here, how it's an aspartate transaminase, you have an alanine transaminase, and so that's what she did. That's one of the enzymes you're going to have here.
Those are both transaminases.
You can have alanine transaminase or some other things, but ALA is going to be the one that, just stick with ALA.
She had PLP and that's B6.
Yes.
Yeah.
So for a lot of these reactions, any kind of, if it ever says transaminase,
That
means
means
PLP.
yeah.
Okay.
Okay, where can we stick that? We'll stick it in our happy note. All right.
So every transaminase?
Yeah, needs.
Yep.
Right.
What's BG?
What?
BG?
BG? That's a 6.
Oh, B6, sorry.
Oh gosh.
I'm just doing it. It looked like a paper, it's fine.
I'm just going to pretend that when I write on paper and it's not a vertical board that my handwriting's beautiful. I'm not going to say that because that would be a lie, but I'm going to pretend it. All right, next, let's see what we got. So we've hit alanine, asparagine, aspartate. We've hit cysteine with methionine. There's a little bit more to the cysteine-methionine pathway. So let's go ahead and add that in there really quick. So if you do happen to draw it out this way, which I suspect most of us have today, you can actually make these pathways pretty close to done just by writing in the enzyme along the arrow that you did to draw between the 2. So this one is going to be phenylalanine. Has something to do with this, either hydratase or dehydratase. Hydroxylase? Oh, you're right. I was the one. Hydroxylase. All right. the OH to the side for my own benefit, so I can remember that it's a hydroxyl. I've gotten that one too many times. All right. So for methionine, what you want to remember is that it goes through the molecule homocysteine. And look, all of a sudden I can spell cysteine. All right, homocysteine. Is homocysteine something that we like to have a lot of in our body? No, it is something that we do not like to have a lot of in our body. And so if this piles up, then we have issues. And so we need, you'll get this later, but we need B9 and B12, folate, and cobalamin in order to process homocysteine into something else. And one of the things that it can end up as is cysteine. And so, and that's the way that we would make cysteine if we wanted it, which has the nice side effect of getting rid of homocysteine buildup.
Can both of those go back and forth, the phenylalanine, the tyrosine, and they can both go both directions?
I can't swear to that one.
Oh,
I don't
so
know
just
on that.
that they're...
I don't think that you can, I'm actually almost positive you cannot go backwards on the phenylalanine for two reasons. One, if you could, then that would mean that phenylalanine would be a conditionally essential amino acid. And number two, I seem to recall her saying that you just couldn't. So
Okay.
one makes sense, the other's memory. Take your pick.
So you're saying you need B9 and B12 to process homocysteine?
Yes, if you're missing either of them, you will have a buildup of homocysteine, and that's why eventually you will, in your nutrition class, you'll have a question that says, how can you tell the difference between a B9 deficiency and a B12 deficiency? Because they both have high homocysteine. And B12 has an extra thing that builds up. Two things build up if you have B12 deficiency, and then only one thing, homocysteine, builds up with B9. But you'll get that later. The other thing, since I know it drives me nuts when someone says another thing, it doesn't say what it is. It's methylmalonyl acid or something like that. It's MMA, and that builds up. So, but that's not for today. All right. So there's those two down. What else do we have? We have Prolene and Serine. Please.
So, she said that it means tetrahydra.
Oh, tetrahydrofolate.
Is it just that H4?
Folate B9 is, yes, that's required for, wait, were you saying phenylalanine to tyrosine? I know that that is connected to the methionine cysteine pathway. I don't remember either way. I don't remember that being involved with phenylalanine to tyrosine, but I'm not going to swear to it. Anytime?
So, but is tetrahydra
It's right here.
biochem the same thing as H4?
She was talking about something else.
No, that was the one above, that was the, for serine and glycine, but this one was a tetrahydrobiopterpin.
Yes, that's different than the B9 thing. So tetrahydromethylfolate
I thought it was just a bio.
is down here. For the,
Yeah.
it sounds like,
But is that, I don't know, I didn't really get that. She just mentioned it, but I didn't get that as being super important.
I have no memory of it, and I don't think that we were asked about that particular cofactor between phenylalanine and tyrosine, but it does look like it exists. It's on the slides, so.
But it's not in red or anything. It's not. She just mentioned it.
So if it's in your head already, keep it, because it's true.
So you're saying if methionine goes to cysteine, you're gonna have, it's gonna clean up homocysteine, right?
Yes.
That's why that's there, okay.
Yep,
Thank
that's
you.
one of the ways that you can clean up
But if it
homocysteine.
didn't, then it would be a bit of.
Okay, at first I was thinking it was an enzyme, but it's not in pink and it doesn't end in A, so never mind.
There you go. It's blue. All right. What else have we got? Looks like
Prolene, I think.
Prolene. All right.
And serine too.
Serine is the one that has a little bit to it. Prolene, I think, was pretty straightforward.
So I have a quick question.
What's
So
up?
going from methionine to tyrosine, is that an OH group that's removed?
It's going to be an OH group that is either removed or put on. I have not looked at
Oh, that's
exactly
the
what
one you
happens. It's
went
a hydroxylase, so it is going to be OH. That's the one that I memorized with water, but it's hydroxyl.
You add a hydroxyl group.
You do? Okay, to make it tyrosine? Alright. Thank you. Alright, so with Prolene, okay, so Prolene, which family of amino acids is the center family for just where all the amino acids are going to have an enzyme to get to and away from? Glutamate. Eventually, most of them are going to have a way to interact with glutamate, and in this case, Prolene is going to join that family. So, this is just naming what the box is. I should not do that. Alright, whatever. That's going to confuse that guy. So, alright, so for this one, I'm trying to see if there are any intermediates that she wants you to know, or if it's just supposed to be that
That's a whole lot of it.
glutamate can go down, and then it can either become ornithine or proline. That's probably what I would remember. You guys remember the urea cycle a little bit? And ornithine, the Henri molecule. All right. So glutamate can either go to ornithine or to proline. And if you have a whole lot of urea in need of, like you have a whole lot of nitrogen that needs to be turned into urea, glutamate will probably preferentially go towards ornithine. And then if you don't have that, where you aren't eating a lot of meat and so you don't have a lot of nitrogen buildup, you're not eating a lot of protein, I should say, then you're not going to need to make as much ornithine, and so you can make proline. I seem to recall that there that's something that can become an issue, is if you eat a lot of protein and so you need a lot of ornithine, then it can be hard to make proline. She's not going to ask you about that, but I know that you guys are fascinated by nutrition because you're in this school. So there you go.
Thank you.
All right, and then the last guy that we're going to hit is serine. So serine has a simple pathway and then a less simple pathway. All right. So you can make serine. There's actually a slide where it shows serine and glycine and then a bunch of molecules together. And so I did the header thing again. All right, stop doing that. All right. Serine, lysine, and then raw materials, which, let's see, that was going to be CO2 and nitrogen. And water. So you can make glycine out of raw materials, and then you can make serine out of glycine. And so that, what was that?
So does glycine break back down to those?
They're
So
both
the two-way-
reversible.
They're both reversible, okay, that's what it is.
Yep.
Okay, we're back to glutamate, to proline, or
Okay.
proline and arginine.
Proline and arginine.
And from arginine, you just make proline. And then you have it.
What are we on right now? I'm so confused.
Sorry.
No, it's okay.
We are on serine. We were going back to the previous one really quick. So we were talking about making serine, which also involves glycine. Now we're talking about glutamate, ornithine, and proline just for a bit. And I think you're right on that one. Let me take a look here. And arginine is one that it's not right here, but arginine is one that can also become ornithine, as that was shown in the urea cycle. So there's more than one way to make ornithine. is what that comes down to. But yeah, I don't think that we use arginine necessarily to make proline. I think it's more just an intermediate of the pathway. But
And I think with serine, what's important there is that it comes from 3-phosphoglycerate.
3-phospho.
Yeah, which is from glycolysis.
Okay.
Yeah.
We
She said
need-
that specifically in class.
And what we need to do is add an amino group to that 3-phosphoglycerate. And in order to do that, the main enzyme is 3-phosphoserine phosphatase, which takes that phosphate off of, I don't know. There's like some steps with the main ones, just knowing that it's a 3-phosphoglycerate, and then without that enzyme, we can't make serine, basically. Is like what she said.
Without the, right.
Without the phosphatate, the
And that leads to this thing
three-phosphoserine.
down here, right? As she said, the Williams bar.
Yeah,
Yeah,
that leads to the Williams, yeah.
Okay, I'm looking at it, and
So the last step where you go from 3-phosphoserine to serine, that enzyme, without that enzyme, we can't make serine.
okay, and that's the one that she highlighted as with the disease.
Yeah,
Yeah.
and just that 3-phosphoglycerate comes from glycolysis, and we just need to add an amino group to that. Is the steps that we do to get to serine, basically.
Okay.
She didn't really like go on the the structures or anything.
The rest of the pathway, just the last enzyme that was the disease-causing enzyme.
Like, you have this and you need to get to this.
Okay.
But you do your thing, I'm just,
No, please.
you know, my notes.
On this one, yeah, I ran short on time, so I'm glad for whatever you have to add to it. We're gonna hack through it together. So,
Yeah, that was the biggest thing that she had mentioned, was the 3-phosphoglycerate. Goes through several steps.
That's what we start with, yeah.
Yeah.
okay, and then the last enzyme, the 3-phosphoserine phosphatase, is the one that causes the disease.
Yes.
Yeah.
All right, then we'll go ahead and write that one up right here, and that'll give it a nice spot that'll make it blend into this picture nicely. 3-phosphoserine
Yeah.
Phosphatase.
phosphatase. Okay. And 3-phosphoglycerate, so we'll go ahead and put glycolysis up here.
Look at
Okay.
us telling you how to do your study.
It's teamwork.
Always works better than one person. Or is it, let's see, like Laskew. When you guys get Laskew, he likes the saying 14 heads are better than one, because he likes to lump as much of the class together as possible.
These are like these slides. Oh my god, for terminology. Do you guys understand the P, N, S, all that stuff?
I've heard of
Oh,
PMS. All
No,
right.
he'll like label a pathogen, it's just a P, so you have to like jog your memory.
Hens, you mean?
No, when Dr.
No, Dr. Laskew,
when he was lecturing.
his first PowerPoint, it was so,
He's much better with like his stuff
oh yeah.
once you get into Gen Med, and so
He is a good teacher.
he is. His slides are better
He's smart.
in Gen Med.
Hmm.
But
That's-
that
Yeah, I don't
first
think
time you
there's
see
anything wrong
him,
with
he wants to give you everything.
Yeah.
It's just like,
Yes,
one
it was very
chance.
broad, for
And
sure.
he wants to ask the questions about the entrance and exit. And
Entrance and
use
exit
kinda
to-
different
Oh, the A
terminology
variant,
for
E
the
variant, yeah.
bloodstream versus lymph and
Let's stay focused, guys.
All right.
Let's stay focused.
And then he puts it on the.
So
Thank you,
this
Amy.
is going to be the other thing that you'd want to stick right here that I would do to remember is, I'm just going to write it in blue because blue apparently is another color. So
He's a good teacher.
WBS, which is Welch-Buren syndrome, if I'm pronouncing that right.
Yeah,
Williams-Buren. Okay,
Yeah.
there we go.
yeah, or just Williams syndrome.
All right, and that's, I think, would kind of consolidate what I would remember on serine if I was back Biochem. All right. So, all of this is well and good to have on a piece of paper, but we want it in our head. So, and I'm going to do this one too, because clearly did not have this part. So, let's go ahead and do alanine, and then Prolene, and then the serine glycine tandem. So let's go ahead and start with alanine, which is a nice place to start. So what does it turn into? There we go,
The
pyruvate.
transaminase.
Yes, transamination with alanine transaminase, ALA. All right, next, go ahead and think about go ahead and think about it first, and then we'll say it. Think about how we are going to make proline. We need proline. All right, how are we going to make proline?
Glutamate.
Glutamate. And just because, what else can glutamate become?
Ornithine.
Ornithine, the ornery molecule. All right, next, with the serine and lysine tandem. First, how can we make it out of nothing? Like out of just bits and pieces? What are those bits and pieces in your head first? And then, after that, what's another way to make serine? So we have two ways to make serine, and we want to hit those. Alright, so the first one is out of carbon dioxide, ammonia, and looks like water I have up here. I don't see that on the slide, so I'm going to do that. Alright, and then, my apologies, and then it turns into glycine, and then glycine will turn into serine. And then the second way to make serine, 3-phosphoglycerate from the glycolysis pathway, and then we use what molecule? Because what enzyme to get it there?
Well, there's a bunch of steps, but the one that you're talking about?
Yeah, the one that is related to the disease.
3-phosphoserine phosphatase.
Yep, 3-phosphoserine phosphatase. And in remembering this one, this is pretty straightforward, but once you look at it, but you have phospho here, you have serine here, so it's phosphoserine. So you just take the first half of this molecule, and then this molecule, and then you have most of the name. All right.
Where do we put the serine and choline though? Where's
What?
choline?
Choline?
Yeah.
I'll have to get back to you on that.
There's 3 ways, right? It can be synthesized. Serine synthesis from serine or choline. Oh, glycine can be synthesized from serine or choline. Never mind. Whatever.
Yeah, I don't have anything on that just
I
now,
don't care.
but hopefully this helped organize some of the information. There is also special products that where you take, it's kind of like a recipe, you take a few amino acids to make something, and for those, I will see if I can make, I actually had a few memory pictures for those, but I'll see if I can make a video or something and put those out so that you guys can have that. And then,
Oh, and that, the serine to glycine is it's a demethylation reaction. I have it in red in my slide.
yes, serine hydroxymethyltransferase.
Yep, that one right there.
I do think that one, serine hydroxy, I would add that one in for sure.
And that also uses PLP, guys.
Yeah. Okay.
And then this one's glycine synthetase, or synthase, excuse me.
PLP.
And both of those are bidirectional. So, all right.
Thank you.
You're welcome.
Is Jen
Glad to do
Madden
it.
here today? Is Jen Madden here today?
No, it's Oriental, and that's the other building. So you guys are free to stay and utilize the room how you will.
What do you do in Gen Med?
Gen Med, to go over a lot of diseases, and basically they say, here's the disease type, here are all the details about the disease, and who tends to get it. Then they move to another one, and you go between 5 and 20 diseases in a 3-hour period. So,
Oh my gosh.
it's fun. It's what we came here to learn, so it's really nice to be
Yeah.
getting the stuff that was what